Hey guys, so we're going to get started with the week 14 review here in just a minute. But if you guys have any specific questions, think about those so that we can go over those at the end. Or if you have any questions about anything that I'm talking about actively, please feel free to ask questions while I'm talking too. That's okay. Especially since on my iPad, which is what I usually get my weekly reviews on, if you have questions in the chat, I cannot see those when you ask questions during the presentation, and so I won't see them until the end. So if there is a question in the chat, just somebody like let me know so that I can open up the chat and look at that. Like let me know verbally, somebody make a noise and let me know, because I can't see that stuff during these things. So I'm just going to check and see really quick. Got 4. All right. All right, so let's go ahead and get started with our week 14 review. We're going to go over our major concepts that we covered in chapters 20 and 21, and then we'll go over questions for those chapters. And then if you guys have any questions about any of the content, and you guys will be able to let me know. All right, so let's start with chapter 20. In chapter 20, we discussed the diseases of the nervous system, and so we specifically got into infectious diseases here. There is no biota in the nervous system. There's none, not bacteria, viruses, or anything. So this is considered a sterile and closed system, and it is an immune privileged site. So immune privilege is a term that refers to the fact that we have a decreased immune response and a specialized immune response specifically for the nervous system to reduce the incidence of any inflammation that could cause damage to the nervous system, especially with our central nervous system. If that nervous tissue gets damaged, that becomes permanent, and so that's not something that our body can really recover or heal from. So it's to our benefit that we have a decreased immune response in the central nervous system, and that it is a closed system. The other aspect of the nervous system that's important is protecting the brain, the central nervous system, because of the blood-brain barrier, where things are less likely to cross over that barrier, including pathogens, but also including any drugs that could be used to treat the pathogens. So then treating disease, if there is introduction of disease into the nervous system, treating that can be a specific kind of problem as well because of the blood-brain barrier. So our first syndrome that we talked about within the nervous system was meningitis, which is referring to inflammation of the covering of the central nervous system, which is the meninges, the dura mater, the pia mater, and the arachnoid mater. And so those layers can actually become inflamed due to infection in the nervous system. And that inflammation is what generates that syndrome of meningitis. And again, we had signs and symptoms that are associated with that syndrome, being like light sensitivity, stiff neck, headache, a fever, and then general, malaise and fatigue associated with that. The 3 different types of infectious meningitis that we discussed on a larger scale is meningitis caused by bacteria, by fungi, or by viruses. And by far, bacteria is the worst kind of meningitis to have. And the most common cause of bacterial meningitis is going to be Streptococcus pneumoniae. We call it the pneumococcus. There is a pneumococcal vaccine available for it. And it causes pretty severe meningitis, and it still has a pretty high mortality rate, but it's not as bad as the meningococcal meningitis, which is the worst kind of meningitis period that you can have. And it is associated with a petechial rash that is very characteristic to that type of meningitis. Neisseria meningitidis is a gram-negative diplococcus, and is transmitted through close contact. And also both of these guys, Streptococcus pneumonia and Neisseria meningitidis, can be part of some people's microbiome in their upper respiratory tract and in the throat. And so whenever those bacteria migrate into the nervous system is when we might see the display of meningitis associated with these. The next one was Haemophilus influenzae, And then there was also Listeria monocytogenes. There's a vaccine for the Haemophilus influenzae serotype B, which we call the Hib vaccine, and that's been very effective in decreasing the numbers of, the prevalence and the incidence associated with Haemophilus influenzae meningitis infections. And so vaccines have been very helpful with that. But Listeria monocytogenes is associated with foodborne illness, and it actually can cause foodborne illness-related signs and symptoms, but could also migrate through the gut and into the nervous system from there, then leading to the development of meningitis. Then we have fungal meningitis. The two causes of fungal meningitis that we discussed were Cryptococcus neoformans, which is usually typically found in bird droppings. It's widely disseminated throughout the environment, but it's more commonly going to cause disease in immunocompromised individuals, and especially people who have AIDS. This seems to be a trademark fungal meningitis that can develop in people that have AIDS. And then Coccidioides immitis, which causes valley fever, and it may lead to the development of meningitis. And then we talked about viral meningitis. Most people are going to recover from this naturally without any treatment within a couple of weeks. It has a very low mortality rate of about 1%. And so this is the least, serious of this. But most viral meningitis is going to be caused by enteroviruses. These are viruses that get into the body through ingestion into the GI tract and then move into the nervous system after that. We also talked about how meningitis can be caused by herpes viruses moving into the nervous system as well. All right, meningoencephalitis. This is when you have meningitis and encephalitis, signs and symptoms displayed. And the causes of the meningoencephalitis that we talked about were amoebas. Both of them were amoebas, and amoebas fall into the category of protozoa. And so our amoeba causes of meningoencephalitis were Naegleria fowleri and acanthamoeba, and they actually cause two different versions of meningoencephalitis. The primary amoebic meningoencephalitis is associated with Naegleria fowleri, and then the granulomatous amoebic meningoencephalitis is going to be associated with the acanthamoeba infections. Both of these guys can be transmitted in fresh water. Typically, Naegleria fowleri is associated with like swimming in like warm freshwater bodies, whereas acanthamoeba is usually found in like tap water and things like that. And it can be transmitted not just by being forced into the sinuses, which is the typical transmission method of Naegleria fowleri, but acanthamoeba can also be transmitted like through the eye, and maybe more commonly associated with that, with the eye infection. It is associated with keratitis as well, which we talked about in the skin and eye diseases. All right, next is encephalitis. So here now we have signs and symptoms associated with inflammation just in the brain and not necessarily associated with meningitis, though many of the encephalitis cases may also lead to meningitis symptoms and signs as well, but it usually is characterized by that inflammation in the brain. The more common cause of encephalitis is going to be the diseases caused by arboviruses, and these are viruses that are transmitted by the biting insects, including ticks and mosquitoes. The more common ones that we're aware of in the United States would be things like West Nile virus and Zika virus. Plenty of people are able to get these infections and not have signs or symptoms. Most people are going to be asymptomatic from many of these arboviral infections, but some people do develop encephalitis when they do have signs and symptoms. We also talked about rabies. Again, it's a viral infection transmitted in the saliva of infected mammals. All mammals can be infected with rabies. And once signs and symptoms appear in the nervous system, it has moved into the central nervous system at that point, and then there's no treatment for that. It is 100% fatal at that point. There are vaccines for rabies available for people and for animals, but it's still prevalent in wild animals despite efforts to try to disseminate the vaccine through baiting practices. And then there was poliovirus. We talked about poliomyelitis, which is a disease caused by poliovirus. It's transmitted through contaminated water. It is an enterovirus because of that, and it moves into the nervous system. And it primarily causes symptoms within the nervous systems associated with the muscles. And so This can also lead to long-term damage to the nervous system and sequelae that can last, including a progressive muscle function degeneration as a result of poliovirus infection. It is mostly eradicated from the world, but it can still be found in Afghanistan and Pakistan, where vaccine efforts haven't been as effective. All right, then we have the prion diseases. Prions are misfolded proteins, and then when they become misfolded, they aggregate in the brain and cause holes to form in the brain tissue. Therefore, these guys cause a disease known as spongiform encephalopathy. Spongiform because of the holes in the brain, encephalo because it's the brain, and then pathy is disease. These infectious particles basically are misfolded and cause like the healthy folded version, the normal folded version, to misfold as a result of that. And that's what we believe is happening whenever these misfolded proteins are spreading the disease and how it becomes transmissible. It's 100% fatal as well, but it takes a very, very long time. We're talking decades for the disease to start to develop once you've been exposed to the misfolded protein. And traditional autoclaving does not eliminate this. It requires a higher temperature as well as treatment with sodium hydroxide. Then we have our two clostridial diseases in the nervous system. This was Clostridium tetani and Clostridium botulinum. These are the bacteria that cause these diseases. Clostridium tetani, again, Clostridium, all species of Clostridium are endospore forming gram-positive rods. And so these endospore forming gram-positive rods, the endospores can be found pretty much throughout the soil, throughout our environment. The same thing for both tetanus and for botulism. Those endospores are found throughout the environment, and then certain conditions will allow them to become vegetative, and the vegetative state is the form that makes their toxins that are associated with their diseases. So these can either enter through breaks in the skin, or in the case of botulism, the more common incidence is going to be whenever the bacteria grows in the food, like canned food. These are anaerobic bacteria, and so they need to be in low oxygen environments. And so when we can the food, if the endospores were not killed off in the canning process, then they will, go through germination and become vegetative cells again and make the toxin in the food, and then you ingest the toxin. Whereas in tetanus, the endospores get into the body and become vegetative and multiply in the body, and then they make the toxin after the fact. So with tetanus, tetanospasmin is the toxin that is associated with tetanus disease, and it leads to spastic paralysis where the muscles can't relax. Whereas in botulism, the botulinum toxin blocks the release of acetylcholine from the nerves, and that's the signal that tells the nerves that they can contract. So by blocking that signal, muscles can't contract. So this leads to a more typical flaccid paralysis. Next, we're moving on to chapter 21, summarizing our diseases of the cardiovascular and lymphatic systems. Blood-based infections, any disease of the blood is called an emia. And so emias, we can define based on the characteristics of the emia. Bacteremia is when any bacteria is in the blood. Blood and the cardiovascular system is, we do not have a biome in the cardiovascular lymphatic systems. There should be no bacteria. There should be no microbes of any kind. And so if there's any presence of any of these microbes in this system, then we would consider that an emia. It's not a normal condition of the body. And so bacteremia is when bacteria are found in the blood and not necessarily multiplying, but septicemia is when the pathogens are actually flourishing and multiplying in the actual blood itself. So if there's transient bacteria in the blood, we would call that bacteremia. But if they are actively multiplying, that's the next level of that contamination. Talking about the actual flourishing of the bacteria in the bloodstream, that's when septicemia develops. Viremia and fungemia are talking about viruses or fungi in the blood, respectively. The first disease that we talked about in the cardiovascular lymphatic system was COVID-19. It's caused by the virus SARS-CoV-2. So SARS-CoV-2 is associated with COVID-19 disease. And so this is a coronavirus infectious disease that came out in 2019. That's why it is named COVID-19. So the virus that causes it is the SARS-CoV-2. SARS-CoV-1 we will talk about in the respiratory chapter, in chapter 22. And we will talk about SARS, which is severe acute respiratory syndrome, but that is a different disease and a different virus than what causes COVID. So COVID can infect many cell types in the host body, which is why we're considering it in the cardiovascular lymphatic diseases, because it is disseminated throughout the body's tissues via the bloodstream and can infect pretty much any tissue in the body. We have different variations of the virus that developed while it was causing widespread pandemic throughout the world, the Alpha, Delta, and Omicron ones. And there are still even more surfacing after that. It causes a wide range of symptoms and mostly starts with respiratory and inflammatory symptoms, but can cause symptoms all throughout the body because it is technically a systemic infection. But 1/3 of the people who are infected are asymptomatic and were likely contributing to the spread of the virus throughout the population. Next, we have the plague. The plague is caused by the bacterium Yersinia pestis. It is a gram-negative bacillus. Its reservoir is in rodents, so rodent populations maintain this. They don't get signs or symptoms from the plague. They just harbor the bacteria and it multiplies in their bloodstream. Then a flea would bite them, and then that flea can transmit it to people. There are 3 different versions: the bubonic, pneumonic, and septicemic plagues. The bubonic plague is associated with infection of lymph nodes themselves becoming infected with the bacteria. So when the lymph nodes are infected, that creates these swollen lymph nodes that are infected and potentially even filled with pus and having an immune response inside of them. Those are called buboes. Then we have the pneumonic plague, where it becomes disseminated in the respiratory system. If this happens, it can be transmitted from person to person and doesn't require a flea to transmit the infection. And then we have septicemic plague. This is whenever we have true septicemia. So the bacteria are actually flourishing in the bloodstream itself. When this happens, the bacteria become widespread, and so does the inflammatory response to it. Because it is a gram-negative bacteria, we have initiation of the inflammatory response by the complement system due to responding to like the endotoxins. So complement can be activated by it, and all of our innate immune cells responding to endotoxin as a PAMP. This all triggers a very high and strong response to endotoxin that we call endotoxic shock. And so this can be initiated from the gram-negative bacteria, as well as the toxins that the bacteria are associated with that can cause damage throughout the tissues, especially in the extremities where we would have death of the tissue and then necrosis of the tissue, and the tissue becomes black. And that's why we call the septicemic plague the Black Plague. It's definitely the deadliest version of the 3 forms of the plague. Moving on to infectious mononucleosis. We just call this mono, or the kissing disease. It's caused by Epstein-Barr virus, which is a herpes virus. It's also associated with Burkitt's lymphoma in immunocompromised patients who aren't able to modulate like their T cell response. And your CD8 cytotoxic T cells can keep certain cancers under control. So the lack of our ability to signal those guys to get rid of the cancers in people who have AIDS, for example, when their CD4 helper T cells, their captains, are being infected by the virus and they have low numbers, they're not able to basically modulate a proper response to cancers like that. And so they're more likely to have the incidence of the cancers. Once you have had a infection with Epstein-Barr virus, because it's a herpes virus, it is a DNA virus and incorporates its DNA into its target cells' DNA. And so once you have mono, you have it forever, like you do with other herpes infections. And it can't even resurface and reinstigate those signs and symptoms associated with mononucleosis infection. Next, we have malaria, a bloodborne disease associated with a protozoa. This is an apicomplexan. Apicomplexans are the types of protozoa that are non-motile. All of the apicomplexans are parasitic in nature, and so they're going to be true pathogens that cause disease in their host, organisms. The protozoa that are associated with malaria are in the genus Plasmodium. And so there's different species of Plasmodium: Plasmodium falciparum, Plasmodium vivax, Plasmodium malariae. And these can often have specific signs or symptoms associated with the specific species of Plasmodium, But regardless, what happens is the protozoa multiply in the blood cells and have their life cycle kind of switching between the blood cells and the liver cells. And whenever they have gone through a life cycle and multiplied in a blood cell, that will then rupture out and release fever and inflammation causing chemicals as a result. And then so the signs and symptoms of malaria typically cycle over 48 to 72 hours due to the life cycle of the protozoa within the blood cells. It's transmitted by mosquito bites. And so the best prevention of malaria is going to be with controlling mosquitoes as well as chemotherapeutic drugs for prophylactic treatment for people who are traveling to areas of the world that are endemic for malaria. We can give them the medications that are used to treat the malaria to prevent them from ever getting the infection. There are vaccines available, and they are effective vaccines, but the vaccine effort is hampered basically by the fact that most of the places where this is endemic, we have a lot of underserved populations and underdeveloped communities that don't have as many as much access to doctors and resources for the booster shots that they would need for the vaccination to be effective. So that's the bigger issue there. So the more of the focus is on mosquito abatement and reducing mosquito populations. Malaria causes crippling cycling symptoms associated with how the protozoa are basically cycling within the blood cells, bursting out of the blood cells, and even moving through the liver tissue. And it can even cause serious brain effects when the protozoa are causing damage in the blood vessels that feed the brain. And so that damage there can lead to coma and neurological damage associated with that. Next were the hemorrhagic fevers. A hemorrhagic fever is defined by the fragility of the vessel walls, especially at the capillaries first, but then this can become causing fragility in the larger vessels as well. And these are all viruses, and some of them are transmitted by mosquito, and some of them are transmitted by blood contact. So the ones that were transmitted by that were not transmitted by mosquitoes and are kept in animal populations, that's where the reservoirs will be, and it's transmitted from the animals to the people without any mosquitoes or anything. That's going to be Ebola and Lassa fever, and then and Marburg as well. And then dengue, yellow fever, and chikungunya are all transmitted by mosquito bites. And these don't always result in hemorrhagic fever. In Ebola and Marburg, yes, it does. This, when you get an infection with Ebola or Marburg, you're going to develop a hemorrhagic fever. But with dengue, yellow fever, and chikungunya, these have other signs and symptoms associated with the fever side of things, with the infection in the bloodstream. And this would be strong shaking, chills, a high fever, and other signs and symptoms of malaise and fatigue and a febrile state. And so those guys can lead to the development of hemorrhagic fever. And that is going to be a much more serious effect of those diseases. But not all of them are quite as serious as like Ebola or Marburg, which is more characterized almost entirely by the hemorrhagic fever caused by that. All right, so next are our non-hemorrhagic fevers. These are cardiovascular diseases. We have infection within the bloodstream, but they are going to have a high fever, which is why they're a fever associated with them, but they are not always associated as characterized anyways by the hemorrhagic side, the bleeding out side. So these are going to be mostly tick-borne diseases, and most of these are bacteria. So Rocky Mountain spotted fever is caused by bacteria Rickettsia rickettsii, and it causes a petechial rash associated with it about 2 to 4 days after the initial symptoms show up in the prodrome. Then the Lyme disease, also transmitted by tick bite, is caused by the bacteria Borrelia burgdorferi. It has a much slower progression of disease associated with it, and it can lead to neurological side effects as well as arthritis due to the inflammatory response to the bacteria. And it has a very typical sort of bullseye rash that is associated with it as well. Ehrlichiosis and anaplasmosis and babesiosis all cause very similar symptoms, and most people will recover from these diseases pretty easily. For those that don't, because the signs and symptoms are so similar between ehrlichiosis, anaplasmosis, and babesiosis, we usually treat them all kind of as one. You have signs or symptoms, you were bitten by a tick. Here are some antiprotozoal and some antibacterial antibiotics to treat like potentially both of those all at once. and we don't usually delineate too much between the two. Ehrlichiosis is caused by the bacteria Ehrlichia. Chafensis and anaplasmosis is caused by Anaplasma phagocytophilium, another bacteria, but babesiosis is caused by a protozoa. All right, so next was Chagas disease. And I'm not even kidding you guys. I went home last night and I was hanging out with my husband on in the backyard, and I saw one of the bugs that transmits the Chagas disease. So that is the kissing bug or the assassin bug, but more scientifically called the triatamine bug. It was just crawling on the our door to our backyard. And these guys, they are biting insects that transmit this. Again, the triatamine bug. They bite and like lap up the blood, and then they defecate out the protozoa Trypanosoma cruzi, and then it gets into the bloodstream by migrating through the skin that way. And it causes an inflamed spot where it enters the skin called a chagoma, and has acute febrile state of feeling ill for a little while, but some people can actually have the chronic response to it, and the chronic response will have chronic inflammation associated with it that can cause cardiovascular disruption, as well as issues neurologically. It's going to be a bigger problem with the Trypanosoma cruzi if it does get to the chronic state. All right, next we have HIV. HIV stands for human immunodeficiency virus. It infects our CD4 T helper cells. Those are our captains. And remember, our captains are CD4 positive T cells. They are going to, the CD4 molecule recognizes MHC2, which is the marker that presents antigen that came from outside of the cell. And only our antigen presenting cells have MHC2. So our antigen presenting cells would be our dendritic cells, our macrophages, and our monocytes, and our B cells. And so they'll pick stuff up from outside and present it on MHC2, and then that will activate our T helper cell. The T helper cell, which was naive and was just a plain old generic T helper cell, then proliferates and differentiates into memory T helper cell, regulatory T helper cell, and then Th1 and Th2. Both of those always come from the response. Then the Th1 helper cells go and moderate our CD8 ones. They will activate that CD8, the CD8 cells. They send signals to them to tell them they can activate. And the CD8 positive cytotoxic T cells, their CD8 molecule recognizes MHC1, and that's the inside antigen presenting marker. And so the Th1 will come over, and whenever the cytotoxic CD8 positive T cell engages with its antigen that's being presented on MHC1, then the CD8 molecule recognizes MHC1, and then the Th1 cell, which was activated from outside via MHC2, that Th1 cell sends the signals to let the CD8 cells become activated. And so that's like a check system to prevent our CD8 cells from just being able to activate themselves. So we're making sure that we're all reacting to an appropriate antigen in the appropriate steps so that we don't have too much damage caused by that. The Th2 cells that were made when our T helper cell went through differentiation, those Th2 cells moderate activating the B cell response, which is our antibody response. But you always have both being made from our T helper cells. All right, 80 ways. So that's our little mini review of CD4 T helper cells. The HIV virus actually infects those T helper cells, those captains that modulate both the inside and the outside response. And so it can actually lead to killing those cells off from the infection. And then we have our significant lower number of our captain T CD4 cells, and they can't moderate our immune response. And that's what leads to the acquired immunodeficiency syndrome, or AIDS, that's associated with HIV infection. HIV virus has reverse transcriptase that allows it to convert its RNA into DNA, and that DNA is then incorporated by the integrase enzyme of the virus into our DNA. It also has a special protein called protease. So what its mRNA codes for one big old long amino acid like chain, and then it's its protein protease will then clip that chain into the individual proteins that'll get packaged up into each virion. And so that's a special protein associated with that virus as well. So we can target all three of these special proteins associated specifically with HIV with drugs that will then only target those. None of these going to be functions that we have in our normal cells. So those are great targets for the drugs, but unfortunately HIV is a master of mutation. And so sometimes we will need multiple of these drugs to keep the viral infections under control. This guy is spread through blood. And so even exposure to other fluids like semen will usually require blood exposure. The only exception to this is going to be with breast milk, and that is usually associated more with the babies that have a lowered immune response anyways. But saliva, sweat, tears, and urine are not considered a vehicle of spreading HIV. The best way to prevent HIV is through barrier methods and not sharing any blood. Blood exchange is the more common way of that getting transmitted. So let's get into our commonly missed topics for these chapters. Back to chapter 20 with our nervous system diseases. This is talking about our immune privilege of our central nervous system, the fact that we have a lowered immune response, we have the blood-brain barrier that prevents pathogens from crossing, as well as a dampened effect of immune signaling within our blood-brain barrier, including decreased amount of MHC1 on all of the cells, and just general signaling molecules and markers associated with inflammation and the immune response. Those are going to be decreased in the central nervous system to act as a protection for that central nervous system. So the blood-brain barrier and the immune privilege aspect protect the central nervous system from infection, as well as overreaction of the inflammatory response. Let's get into our questions. So we want to keep in mind, we have the blood-brain barrier, prevents things from crossing over, and we have immune privilege. Decreased immune response. This is a decrease in the cells and the signaling involved. So our first question is, which of the choices best describes the function of immune privilege in the central nervous system? Does it get the strongest immune response possible? No, the whole idea is that it has a decreased immune response. So it's not going to be A. Does it have no immune response? Again, it has an immune response, but it's just decreased. Does it have a lowered immune response and specialized cells to control and prevent damaging effects of inflammation? This sounds more like what's going to be our answer. Or does it have its own lymphatic system? It does not. It's just a controlled version of our own immune system, as well as certain specialized cells that are designed just to be operating with the immune function in the brain tissue. Our next question is, what role does the blood-brain barrier play in achieving immune privilege? So our blood-brain barrier prevents things from crossing over into the brain and the central nervous system. And so that's the whole function of the blood-brain barrier. And an immune privilege is associated with a decreased immune response. So let's go through our choices and see which one lines up with that line of thinking. Does it allow immune cells to more freely move? I think that's what I was supposed to say, to move freely into the area to protect the area more effectively. It's not this one. We're not allowing them in there more. We're going to be allowing them in there less. Does it prevent the entry of pathogens into the bloodstream from the CNS? So this is the issue. The blood-brain barrier really is there to prevent pathogens moving from the blood into the central nervous system, not the other way around. Does it restrict the passage of immune cells? It does. It definitely restricts. It restricts passage of immune cells and pathogens and drugs potentially and other substances from the bloodstream into the CNS. This one is lining up pretty well. Does it enhance the immune response? No, it's not going. The blood-brain barrier doesn't enhance the immune response. It prevents certain immune cells and certain immune signaling molecules from crossing into the blood-brain barrier so that we can make sure that we don't have a heightened inflammatory response to protect our central nervous system. So C is our answer there. Next, we're going to talk about our fungal meningitis. Fungal meningitis is caused by fungi. Fungi are eukaryotes. What's a eukaryote? It has a nucleus and membrane-bound organelles. like mitochondria, the endoplasmic reticulum, the Golgi apparatus, vacuoles, lysosomes. All of those things are membrane-bound organelles. So eukaryotes have that. Eukaryote means true nucleus. And so these are fungi. And so that's one way that we could distinguish the fungi from the bacteria is by looking for cells that have membrane-bound structures inside of them. So our two causes of fungal meningitis are going to be Cryptococcus neoformans and Coccidioides immitis. Again, Cryptococcus neoformans is a fungus that's found pretty ubiquitously throughout the environment, but is more concentrated and more commonly found in bird droppings. And it's usually only going to be causing disease in folks that are immunocompromised. And this is more closely associated with folks who have AIDS. This is very common in the homeless populations. And Coccidioides immitis is found again just in the environment in dry regions in the deep Southwest, and it will cause mostly a respiratory illness and maybe widespread febrile state, but it can move into the nervous system and cause meningitis, especially in immunocompromised people, of course. All right, so we have the two fungal meningitis. Again, fungal meningitis is caused by fungi. They are eukaryotes. And our two options were Cryptococcus neoformans and Coccidioides immitis. So again, our first question, which of the following is a causative agent of fungal meningitis? So here we're going to be looking for fungi. We have Clostridium perfringens. This is a bacteria associated with gas gangrene. And we will learn soon it's also associated with food poisoning. It's not this one. Then we have Cyclospora. Cyclospora is a protozoal cause of diarrhea. We'll learn about that in chapter 23. Coccidioides immitis, here we go. And then Crimean Congo HFV. HFV is hemorrhagic fever virus. I just put this one in there as a random outlier. So this is a virus associated with hemorrhagic fever, which is one of the ones we don't have to know for our test, just so you know. But it is one that I studied in graduate school. Random outlier, I just picked some C names. All right, so Coccidioides immitis was our fungal one. And again, remember that the other fungal cause of meningitis was Cryptococcus neoformans. So which form of fungal meningitis is usually associated with AIDS? People who have AIDS are going to be more likely to have this. So again, diarrhea, protozoa is not Cyclospora, and we can also knock out Clostridium perfringens, which is a bacterial, which we had over there. Then we have Cryptococcus neoformans and Coccidioides immitis. Both of these are fungal causes of meningitis, but the one that is more commonly associated with folks who have AIDS, especially in our homeless population that are living exposed to bird droppings, this is going to be Cryptococcus neoformans. Coccidioides immitis is not as commonly associated with this. Again, Coccidioides immitis usually causes valley fever. All right, moving on to our PAM and GAM. Both of these are amoebic meningoencephalitis. PAM is primary amoebic meningoencephalitis, and GAM is granulomatous amoebic meningoencephalitis. And we do need to know that they are different. You don't necessarily know about what characteristics define each, but you do need to know the causes for each. And so PAM is caused by Naegleria fowleri. And then acanthamoeba causes GAM. Both of these have a very high mortality rate. We're talking 97 to 98% mortality rate. And even people who survive these infections are going to have deficits as a result of them. So there's more information on these diseases if you want to learn more information, but I'm going to move on to the questions. So PAM and GAM, primary and granulomatous amoebic meningoencephalitis. PAM is more commonly associated with Naegleria fowleri, and then GAM is associated with acanthamoeba. So which of the following pathogens is an amoeba? Neisseria gonorrhoeae. This is a bacteria. We're going to learn more about Neisseria gonorrhoeae in chapter 24 in our genitourinary diseases. This is a sexually transmitted disease. This is gonorrhea. But we've already talked about one species of Neisseria that was a bacteria, and that was Neisseria meningitidis, also in the same chapter. So it's not this one. Alcaligenes faecalis. Again, I don't necessarily want you to know this one off the top of your head, but this is one that we talk about in the lab. This is a bacteria. If you had a question like this on your test, they will all be organisms that were on your study sheet, specifically on your study sheet for lecture. Okay, so not this one. Naegleria fowleri. Naegleria fowleri is going to be our amoeba, causes PAM. And then we have erythema infectiosum. This is talking about the organism that causes hand, foot, and mouth disease. This is the disease associated with that. This is a viral infection. Again, this one and this one weren't on your study sheet. I wouldn't put that, but I reserve the right to put Neisseria gonorrhoeae. I reserve the right to put Neisseria meningitidis or Streptococcus pyogenes or Streptococcus pneumoniae or Yersinia pestis or any of these other organisms that I would expect you to know at the very basic thing that they are bacteria or a protozoa or a fungus or a helminth or a prion or a virus. At the base level, that and what disease they're associated with, you will be expected to know those things and differentiate between them. So which of the following pathogens is the cause of PAM? PAM is associated with Naegleria fowleri. Acanthamoeba is associated with GAM, so it's not this one. Giardia lamblia is a diarrheal disease, and Trypanosoma cruzi is a cardiovascular disease transmitted by that kissing bug. That was Chagas disease. All right, our next aspect of chapter 20 is our arboviruses. These are the viruses that are transmitted by biting insects. There's a whole list of a whole bunch of diseases that fall into this category. The most common ones are going to be West Nile virus and Zika virus, but there are other ones that do occur in the United States. So I'm going to move on from this. So I'm going to keep our little note up here, arboviruses. Viruses that are transmitted by biting insects. Especially mosquitoes. All right, so what is and these guys are associated with encephalitis. Inflammation of the brain. So what is a primary transmission method of arboviral encephalitis infections? Again, biting insects like mosquitoes. They're not airborne. They're not transmitted by fomite. A fomite is like a non-living inanimate object that like a living creature contaminated it, and then another creature contacted that contaminated object and got sick from it. So if I sneeze on my keyboard, you use my keyboard after me and you get COVID from me through the keyboard is a fomite. And then transplant tissues, no. This is associated with mosquito bites. All right, because they are arboviruses transmitted by biting insects. So what is encephalitis? Itis is going to be inflammation. Encephalo means brain. So let's see what we can see. Is it inflammation of the skull? No, that's the bony covering. Is it infection in the nerves? We're not talking about all the nerves. We're talking about specifically the brain tissue here. Is it inflammation of the brain? Yes. Is it infection of the brain? So this is not correct. This is not correct, okay? You can have infection in a brain without inflammation, potentially. And infection in the brain can cause inflammation. Inflammation in the brain is called encephalitis, and you can have inflammation in the brain from things other than infections, like trauma or exposure to certain chemicals, for example. So C is our answer. It's specifically inflammation of the brain. All right, next moving on to rabies. Rabies is a viral disease. It's transmitted through the saliva of infected animals, and this is primarily found in mammals, and all mammals can potentially have rabies. I literally had a student in my evening class, bless her heart. She we were talking about the rabies thing, and she came into class that morning before we talked about rabies, and she's like, man, I had this squirrel like bit me. Apparently her dog attacked the squirrel, and she was trying to rescue the squirrel, and the squirrel bit her. And we're assuming the squirrel bit her because it was agitated and angry from the situation. But the problem is, what if the squirrel bit her and it had rabies? If the squirrel bit her and had rabies, then she could have gotten rabies, and she's not going to know she got rabies until she has signs and symptoms. And at that point, there's nothing we can do for you. So she ended up going to the ER, and she's getting her series of shots and immunoglobulin infusions now. Like literally, we just discussed this yesterday. So that's fun. Gotta love it. But better safe than sorry, honestly. Did the squirrel have rabies? 99% chance no, but in that 1% chance that it may have, better safe than sorry. If you want to die from the rabies, then don't get treated for it. And so that's what the issue is there. So anyways, this is, let's move on to the actual questions about it. There's more information on that slide if you are interested in more information about rabies. But which type of pathogen is the causative agent of rabies encephalitis? Rabies, again, is caused by the rabies virus. This is a viral infection. And which of the following is not included as a way to prevent rabies encephalitis? Do we give post-exposure prophylactic vaccination and immunoglobulin? We do give that. Do we give pre-exposure prophylaxis vaccination? You can do that. We do that for our animals and for our veterinary technicians and the vets themselves, people that are going to be around animals that could have rabies, as well as just our pets and things like that, preventing that, not needing the exposure. Do we try to control rabies within our animal reservoir populations? Yes, we do. Do we give antibiotics for rabies? Antibiotics are for bacterial. This is our answer. This is a viral infection. You will not treat viral infections with antibiotics. All right, moving on to our cardiovascular diseases. First is the plague. Again, it is caused by the bacteria Yersinia pestis. This is a bacteria that is gram-negative bacilli. Its reservoir is found in rodents. It's transmitted by fleas, and it causes 3 different forms of plague: bubonic, septicemic, and pneumonic. So flea bites transmit the bubonic and the septicemic, whereas pneumonic can be transmitted through respiratory droplets from person to person. And then we have specific signs and symptoms associated with these. The bubonic plague is infection in the lymph nodes, and the lymph nodes become swollen from the infection. That's a bubo. In septicemic plague, you won't have the lymph nodes being affected, but you have infection within the bloodstream that can lead to septic shock. And then in pneumonic plague, we have the lung. It's highly contagious to the airways. Through the airways, this is more of a respiratory disease then. So let's move on to the plague. Again, plague, we will have 3 kinds. We have bubonic, the lymph nodes, pneumonic, the lungs, respiratory infection, and then septicemic. Like septicemia, we've got a blood infection here. All right, and it's caused by the bacteria Yersinia pestis, gram-negative bacteria. There's a little info to refer to when we're answering our questions. Which of the following choices represents the type of plague referred to as the Black Death? The Black Death resulted from septicemic plague. This is because the spread of the bacteria in the bloodstream would lead to necrosis in the vasculature and in the extremities as a result. So the necrosis led to more necrosis from the bacteria spreading into those tissues and led to a blackened color of the tissues that were affected. That's associated with the septicemic plague. Which of the following choices represents the type of plague involving infected, inflamed, pus-filled lymph nodes? So up here, we can see that the lymph nodes one was our bubonic plague. Pneumonic is in the lungs. Cutaneous would refer to a skin infection, but that's not one of our choices for the plague. And septicemic is our bloodborne version. All right, next is malaria. Malaria, again, is transmitted. It's a protozoal disease, and the genus of that is Plasmodium. We have Plasmodium falciparum, Plasmodium malariae, whatever, but its genus part of that is the Plasmodium. And this is an apicomplexan protozoa. Means it is non-motile and parasitic. We can control the spread of malaria through prophylaxis. So this means using drugs to prevent the infection. Vaccination is possible, and we also have mosquito abatement. So removing standing water, using insecticides, using mosquito nets or screens on doors, and using genetically modified male mosquitoes that can't reproduce to reduce the populations that way. It has crippling, cycling symptoms that are associated with the protozoa's life cycle in the blood and in the liver. All right. So malarious protozoa. Called Plasmodium. It's the genus name. And again, this is a bloodstream infection. They live inside of the blood cells and liver cells. So which type of pathogen causes malaria? Is it a bacteria, an archaea, a eukaryote, or a helminth? Well, first of all, helminths are eukaryotes, but it's not even a helminth. It's a protozoa. So helminth isn't the right answer because it's not a helminth, literally. A protozoa is a eukaryote. Bacteria and archaea are not eukaryotes. They are akaryotes. So our answer here is eukaryote. How is malaria transmitted? It is a bloodstream infection transmitted by mosquito bites. Next, we have Lyme disease and Rocky Mountain spotted fever. These are tick-borne diseases that a lot of my students get confused on the exam if I have them in a question together. Rocky Mountain spotted fever is caused by Rickettsia rickettsii, and Lyme disease is caused by the bacteria Borrelia burgdorferi. Rocky Mountain spotted fever, R for Rickettsia. Rickettsia. Man, anyways, at least you guys don't have to write it out. All right, so these are both bacterial diseases for Lyme disease and for Rocky Mountain spotted fever. Again, Rocky Mountain spotted fever, Rickettsia rickettsii, and then Lyme disease is Borrelia burgdorferi. Ehrlichiosis, Ehrlichia, babesiosis, Babesia, anaplasmosis, anaplasma, but Rocky Mountain spotted fever and Lyme disease, these aren't in their names, right? And so that's why I'm trying to remind you guys, Rocky Mountain spotted fever is caused by the R bacteria for this one, all right? Again, they're all tick-borne diseases. So if I ask you guys, which disease does Borrelia burgdorferi cause? The other tick-borne diseases had it in their name, except for Lyme disease and Rocky Mountain spotted fever. But the R in Rocky Mountain spotted fever says that this is the one that's associated with Rickettsia rickettsii. And Lyme disease would then be the one that's caused by Borrelia burgdorferi. There's no disease called borreliosis. It's Borrelia, whatever, it's right down here. This is Lyme disease. Rocky Mountain spotted fever, Rickettsia rickettsii. Babesiosis is caused by Babesiosis protozoa. Ehrlichiosis is caused by Ehrlichia chafensis, which is a bacteria. This is also bacteria, and this is also bacteria. They're all tick-borne diseases. Which organism causes Rocky Mountain spotted fever? This is Rickettsia rickettsii. And we know this because of the R in Rocky Mountain spotted fever will remind you that that is that. Borrelia burgdorferi is Lyme disease. Anaplasma phagocytophilium is anaplasmosis. Ehrlichia chafensis is ehrlichiosis. All of these, again, are tick-borne diseases. All righty, so that is it for our review for week 14. Do you guys have any questions for me? Okay, if you guys don't, then I'm going to call it quits for the day. And you guys know you can always email me if you have any questions when you are studying. And happy studying, and be sure you get your lab unknown reports done. Earlier, the better. The sooner I can get back to you with feedback. And otherwise, I guess I will see you guys next week for the lab reviews. You guys have a good weekend.